Our poster is about the role of Ime4 in Drosophila spermatogenesis, which falls under the relatively new field of epitranscriptomics. Our lab is looking at the M6A modification in RNA, which is the methylation of adenosine in the N6 position. This is important due to its connection to a multitude of cancers and viruses. M6A is added by the enzyme Ime4. In our experiment, we look at Ime4 in Drosophila mutants. We use Drosophila as they are a well-characterized model organism. Their testes make good models for immunostaining due to their transparency and clearly laid out stages, making recognizing abnormalities simple. In testes with abnormal amounts of functioning Ime4, a tumor-like phenotype was observed as a swollen tip in the testes. This phenotype has also been recognized in an abnormal amount of TSN, which is a protein that is needed to silence transposons. Transposons are genes that jump around which can cause harmful mutations when not silenced, like cancer. From the observed similar phenotypes, our lab decided to do further research on a possible connection between Ime4 and TSN.
To look at this connection, we looked at the RNA sequence of TSN and if it has Ime4 binding sites, which have been found to include AAA-CA and AAA-CC. Having these binding sites makes this relationship possible, and TSN does include these sequences, highlighted in Figure 2. Having these binding sites, however, wasn't enough to show the relationship, so we looked at the most probable hairpin structures that form within the RNA because Ime4 needs a secondary structure to recognize its target. The most energetically favorable structures are shown in Figures 3 and 4 for each binding site. We then quantified the effects of Ime4 deficiency using RT-qPCR, which shows RNA expression levels. The results suggest a potential direct relationship where a lack of Ime4 causes a decrease in TSN expression, which corresponds to upregulation of transposons copia and 412. To visualize the quantity of Ime4 seen with TSN, we counted spermatids in the testes due to a positive correlation in the amount of sperm in TSN, and to see if there was a change within the controls in homozygous mutants. We stained the testes with DAPI to show all the cells in Figure 6, and from the results so far, we haven't seen any significant differences.
Although there was no significant difference found in the amount of spermatid bundles between the controls and homozygous due to the other evidence such as binding sites, hairpin structures, and the significant decrease in TSN, and upregulation of transposons in Ime4 deficient mutants, it is still highly plausible that there is a conserved mechanism where Ime4 regulates TSN and prompts for further research.
If a conserved mechanism is found between Ime4 and TSN, we can gain a further understanding of how M6A functions, which can increase its medical applications in humans.